like James said, welcome to JBay, which is short for Joint Bioenergy Institute. So JBay consists of Lawrence Berkeley National Laboratory, Sandia National Laboratory, UC Berkeley, UC Davis, the Carnegie Institute of Science, and Lawrence Livermore National Laboratory. So our main focus in JBay is to improve the conversion of cellulosic biomass into biofuels. And cellulosic biomass is the main component of plants. And according to the U.S. Department of Energy, cellulosic biomass is a part of the plant that's fibrous, woody, and generally inedible. So JBay is split into 4 departments. It's feedstock, deconstruction, fuel synthesis, and technology. So in the feedstock department, they mainly focus on engineering new plants so it could be easier broken down into simple sugars. Deconstruction, which is us, they focus on improving enzymatic breakdown of cellulosic biomass. Fuel synthesis, they focus on producing new biofuels using synthetic biology. And the technology department just focus on building new technology that can further help the research. So fossil fuels. So according to this picture, the United States relies on fossil fuels more than any other country in the world. And also we emit the most carbon dioxide than any other country in the world. So according to Dr. Jeff Dukes from the Carnegie Institute of Washington and Stanford, it takes 20,000 kilograms of ancient plant matter to make 1 liter of gasoline. So this just means that we're wasting more fossil fuels than we are actually creating. So therefore, we need a new source of transportation fuel. Oh crap! Why'd I go back? Oh yeah. So, according to the Department of Energy, cornstarch-derived ethanol is a primary source of biofuels in the United States. So, cornstarch, as you guys can see from here, cornstarch is highly branched, or amylopectin, which consists 75% of cornstarch, is highly branched. And whenever an enzyme comes in and tries to break it down, it has more ends, like these are the ends, to grab onto. So it has more ends to grab onto, so it breaks it down faster. So the pros and cons of cornstarch. So advantage of cornstarch is that it reduces greenhouse emission by 21.8%. And according to the Iowa State University, corn-derived ethanol lowered gas by 29 to 40 cents. And as I previously mentioned, it's easily broken down and converted to biofuels. One of the disadvantages of cornstarch is that the cultivation of the fields increases the CO2 or carbon dioxide emission. We have to use a lot of water and fertilizers to cultivate the fields. And corn-derived ethanol, it cannot be transported by pipes. It has to be trucked. So, and one of the main disadvantages is that it's causing hunger in many developing countries because of the food prices are rising because we're using corn as a source of biofuel. So cellulosic biomass. So cellulosic biomass is the main component of plants, and it is also the most bountiful waste product in the United States. The current supply from agriculture and other sources is about 1 billion tons a year. So large, as you guys can see here, large amount of the plant cell wall is composed of lengthy chains of sugars, or also known as polymers, which can be converted into biofuels. So, as you guys see here, it has 3 main components. Cellulosic biomass has 3 main components. It's cellulose, hemicellulose, and lignin. So, so cellulose is hard to reach because it's surrounded by hemicellulose, and the hemicellulose is linked together by lignin, and lignin acts as a barrier to prevent the enzyme to attack it and break it down. So cellulose is the most overflowing biological material on Earth. So converting cellulose into biofuels has been an important notion in recent years, but due to its structure, it hasn't been as efficient. So another reason for that makes cellulose so hard to degrade is because of its structure. If you guys see, look at this picture, the monomers that make up these cellulose chains are stacked on top of each other, so this makes it a tough and largely insoluble material. So this just means that enzymes can't break it down as efficiently. So if we compare this to cornstarch, it takes 7 days to convert cellulose into biofuels, as cornstarch takes 2 days to convert into biofuels. And it also takes 100 times more enzymes to convert cellulose into biofuels as it does to catalyze the same conversion of cornstarch. So this shows the whole process. So we start from the trees, the plants, we harvest it, we add acids and heat it up to make the cellulose available to the enzymes. So after we get the cellulose, we attack it with enzymes, break it down into sugars, and then insert the sugars into microbes, and then we get biofuels, which is put in our cars, into trailers and airplanes. So an advantage of using cellulose is that the carbon dioxide that is released, it is collected from living plants. So when we burn it, it releases carbon dioxide, but that's carbon dioxide that's already in the environment. And like fossil fuels, we have to dig it up from the ground and bring it up again. So since our current methods of degrading cellulose is not as efficient, we are searching for an unknown cellulose degrading enzyme from the environment. So our summer project mainly focused on improving this part of the cycle. So it focused on improving the deconstruction process, which is this part. So the first step of our process is to isolate the cellulose degrading bacteria to get a single colony, or a pure colony. So now I'm going to pass it down to Ian, which is going to talk more about it.
Let us know if you guys have questions throughout, and we'll try to answer at the end. We'll probably pass them off to Clem.
Hello, my name is Ian, and my research question is how do we isolate bacteria? As Martine just explained, cellulose is structured in a way that is very hard to break down. And we do know that there's microorganisms that can break down organic compounds. The microorganism we're going to use is bacteria. Why bacteria? Well, they're single-celled organisms. They produce enzymes, which are proteins that can speed up chemical reactions. They're very easy to manipulate, so we can get them to do what we want. They degrade organic compounds, and they grow very rapidly, and they're everywhere. What do we want to do with the bacteria? We want to isolate the bacteria so that the certain bacteria that we use, we're going to isolate it on a cellulose so that the bacteria will be forced to break down the cellulose by creating enzymes. You can think of it as you have a room full of meat and you want to get rid of the meat. So you put a vegetarian in there and lock him in there. The way he's got to survive is he's got to eat the meat. And that's what you want to do. Well, places where we find bacteria are compost piles, trees that are degrading cellulose, and forests, like forest floors, and anywhere where cellulose is being degraded.